Good. All right, let's see. So spring 2023, I put the date in there. And it was the wrong semester. So what you've got in mammals, there is a genetic difference. So we've got 23 pairs of chromosomes. The 23rd pair is referred to as the X chromosomes. They are different whether you're male or female. Females have what is in fact a true homologous pair. They have two homologous chromosomes. They call them the X chromosomes. Again, homologous chromosomes have the same genes in the same location. They can just have different versions. The male, that last pair of chromosomes, the sex chromosomes, they are different. While this in meiosis acts like a homologous pair for cell division, this is not truly a homologous pair. The X and Y chromosome do not have the same genes in the same position. In fact, if you look at the Y chromosome, do you think there's any genes on the Y chromosome that are essential for normal, in quotes, physiology? No, there can't be. Because if there was, how can you be dead because you don't have a Y chromosome? So there can't be anything essential on it. So what it has, of course, I'm not a molecular geneticist, but it has a lot of stuff. We don't know what it does. It may just be sort of residual remnant genes that no longer function, but a lot of it doesn't seem to code for any proteins. But there is one gene on it. It is called, I'm going to call it the HY. The term's about that. It's not called the HY antigen anymore. That's a term for some other mammals. It's just the only name I can remember off the top of my head. But this is a gene. So there's a gene on the Y chromosome. When that gene is expressed, that is what will lead to the development of male characteristics. So let me switch to an early embryo. If you look at this top picture here, this up here, it's about a 5-week embryo. This is sometimes referred to as the indifferent stage. All of the structures here are present to make either male or female structures. The gonad, what they're calling is gonadal ridge, that has tissue in it that can make the ovary, or it can make the testes. They're both there. The kind of, what is that, teal, greenish, blue-looking color there, the mesonephric ducts, they will make all the male structures. Think vas deferens, seminal vesicles, that sort of thing. These kind of purple tubes coming up here, the Mullerian ducts they're called, those can develop into all the female structures, uterus, uterine tubes, and so forth. So they're all present. I mean, if you took an ultrasound of this 5-week embryo, they couldn't tell you if it's a boy or a girl. It's both. It's got everything. So that's kind of how you start off. Now, what happens at this point right here, right about 5-6 weeks, a gene on the Y chromosome will be expressed, and all it does, it targets this indifferent gonad, or this gonadal ridge, and what it does, it makes the parts that are going to develop into a testes remain, and it stimulates them. The parts of that indifferent gonad that would be ovaries degenerate. That's it for the genetic difference. This is the only step you need. If that Y chromosome is present, you'll get this protein produced. That causes the gonad to develop into a testicle. That's it. That's it genetically. Everything else is still the same. So now you have testes. Well, what do testes do? Testes, when they develop, they will secrete testosterone. And it turns out, if this individual is a male, and the gonads develop into a testes because of the presence of the Y chromosome and this HY antigen, then the testes, prenatally, before birth, they will crank out, basically a bunch of testosterone will be released. So, if testosterone is released, that's the key. If testosterone is present, development will follow this path. And basically what it causes is those mesonephric ducts are retained and stimulated. They develop into the epididymis, vas deferens, seminal vesicles, and so forth. All those Mullerian ducts, kind of shown with the dotted lines here, they degenerate. They're broken down. So the female structures degenerate, and you end up internally getting testes forming all the male ducts and so forth. If there is no testosterone, that's kind of the default. If this were female, this wouldn't be present. The testes don't develop, they degenerate, the ovaries develop. Ovaries don't produce testosterone, there's no male sex hormone, and so you'll retain the Mullerian ducts, they'll develop into the uterine tubes, uterus, and so forth. The male structures, the mesonephric ducts degenerate. You have female structures internally. So really development, it's the hormone. I mean, yeah, you need that genetic difference to get the gonad to develop into testes. But once you have that, that's it, the genetics. The rest is endocrine. This shows you the external structures, and here's where I'll go through some of the homologous structures. Again, same stage embryo. I'm going to start here, and then we'll just kind of see what happens. If this is a genetic male, you get testosterone, male structures, no testosterone, kind of the default is a female structure. So kind of the bits we'll look at, let's see if I got enough pens here. They've color coded a few bits. So kind of this piece of yellow up here, that is called the genital tubercle. A tubercle is just a bump, it's just this little bump of tissue. Then on either side, you will see kind of a couple of ridges of tissue. I'll put it in red, they're kind of purple on that diagram. These here and here, the large C, those are urogenital folds. And there's a gap in the middle. That's the urethral groove. So this is an open spot here. It's not labeled. That's the urethral groove. And then if you move laterally a bit out to the side, these big blue blobs there, those are the labioscrotal swellings or folds. So they're out here. They call it folds. So that's how we all start. Now, internally, what's happening at about this point, that's when you get the Y chromosome expressing one of its genes, and the testes develop. If the testes develop, then testosterone will be secreted prenatally. That's kind of the key. If there is testosterone, this is what's going to happen. You follow these parts. If you look at the genital tubercle and you kind of follow it down in development, in a male, that genital tubercle, that'll be the glans penis, the head of the penis, is what the genital tubercle ends up forming. The urethral folds right here, they elongate and they fuse together. This urethral groove will actually seal shut. It kind of grows shut like a zipper. The urogenital folds elongate, fuse shut. That's going to end up forming the shaft of the penis. So the urogenital folds fuse together. That's the shaft of the penis. And then finally, you can probably guess, the labioscrotal folds. That's what the testes will eventually descend into. That's going to be the scrotum in the male. That's what happens if testosterone is present. If there is no testosterone, you follow a different path. If you follow the genital tubercle of a female, no testosterone, that ends up being the clitoris. So those are homologous structures. The genital tubercle, that's the same embryonic structure. In a male, it ends up being the head of the glans penis. In a female, it's the clitoris. So the clitoris and the glans penis are homologous structures. They start off as a same embryo. The urogenital folds, well, it forms kind of the shaft of the penis in the male. It doesn't elongate as much. They don't fuse closed. Those end up being the labia minora. So the shaft of the penis, labia minora, those both develop from the urogenital folds. Those are homologous structures.
So you have no testosterone.
If there's no testosterone, this is what you get. The urogenital folds will end up developing into the labia minora if there's no testosterone. Testosterone, it's a shaft of the penis. And then finally, the labioscrotal folds in a female, they just end up becoming the labia majora, as you could probably guess from the name. So this is all hormonally driven. The key is there's a prenatal surge or release of testosterone. That's what makes all the male reproductive structures develop. But then that testosterone goes quiet, as we'll see in a bit, doesn't come back into puberty. But you need that prenatal release of testosterone. That's what causes, so it's hormonal. It's not really genetic, although genetics kind of sets it in motion. Once you get the testes, development of everything else is due to the evidence of the hormones. And just to kind of illustrate this, I'll give you an example here. Draw a picture. This is a cow. All right, now I'm not a rancher, so if I say something wrong, it's all right, you can correct me. But unlike humans, this will not happen in humans, but in cattle, all the mammals have the same system. Sex is determined whether you're a rat, a cow, a dog. It's all the same. The timing of the events is a little different, but everything's the same. But cattle have a placenta that doesn't quite work the same as humans. So this would not happen in humans, but this is good to demonstrate this effect. In cattle, they'll often have twins. So let me draw the uterus in here. One baby. Two babies. So you got twins here, a couple of calves. But let's say one of these is a male calf, one of these is a female. Now they'll go through the same steps as a human. Again, I don't know the timing for cattle, but they have it in a different stage where the males and females are the same. Then at that point where the Y chromosome is expressed, alcoholic gonads develop. So it's the same process. Again, I don't know the timing, the process is the same. The difference here is in cattle, a placenta is much leakier than it is in a human. So what happens when this male hits that what's equivalent to 6-7 weeks of development in a human, that male will start cranking out testosterone, which is going to control its development, cause penises and such to form. But some of it leaks. So because they have, again, I'm going to refer to it as a leaky placenta, things can move across easier than they do in a primate. So the testosterone will get into the mother's blood, and it will be delivered to the female calf. And what you get in extreme cases, I think they call these free market twins, I'm not really sure. These two twins can be born, you look at them, penis, scrotum, they both look like males. One of them's a genetic female, but the testosterone just from that other male calf leaks across and it completely controls the development of the female, masculizes it. I mean, you couldn't necessarily tell the difference. Look, typically they're sterile, so they don't reproduce, so they end up being breakfast earlier than normal, but it's the same process. So it's the testosterone that has a huge impact on development. That's why, and again, humans, you don't get this. We don't have leaky placentas when there's twins. It's not, it's not a big issue. But you can get weird things happening. Like, for example, if there's a mutation, let's say a male, what's going to be a male, XY, let's say the receptors for testosterone don't function. That individual will be a genetic male. The gonads will develop, but because there's no receptors for testosterone, none of the tissues respond. So you can get situations where an individual is born, looks female, has all the female parts, except internally it's got testes, but genetically it's a female. Again, it's the hormones that cause the production of the primary reproductive structures as well as the secondary sex characteristics as well. All right, that's the end of the development part.